Hello and welcome to my channel Lab Easy, where we will be discussing various techniques which are commonly used in biological sciences labs all across the world. So, today we will be discussing Western blot. So, Western blot is a very routinely used technique for detection of protein samples for analysis of the protein of interest. So, there are three main steps in Western blot: separate, transfer, and detection. So coming to the first step of Western blot, that is separate. So here, why do we want to perform Western blot in the first place? So for example, we are growing a particular cell where we want to observe whether my protein of interest is being expressed or not. So for which we need to perform a detection method by which we'll be able to detect the presence of the protein, and that method is Western blot. So suppose we are growing a particular cell and then we take the lysate of the cell. That is, we lyse the cells and take the proteins in the supernatant. And then what we do, we perform gel electrophoresis. So why do we perform gel electrophoresis? So that our proteins, like the whole cell proteins, can now be separated according to their molecular weight. So before performing gel electrophoresis, if it is a denaturing gel electrophoresis, so what do? We treat the protein sample with SDS. So this comes under sample preparation. So we treat the protein sample with SDS. SDS is sodium dodecyl sulfate, which not only denatures the protein but also imparts uniform negative charge to the protein so that the protein can be separated on a polyacrylamide gel according to its molecular weight. So this polyacrylamide gel is prepared. A typical gel cassette looks like this, and then this sample is loaded onto the wells of this gel, and then this gel is placed into this cassette and placed into the buffer tank. So the running buffer of SDS page is TGS. TGS stands for tris glycine and SDS. And then this gel is run in this apparatus by the application of a certain electric current. So this is the process by which we can separate the whole proteins of the cell by their molecular weight. So now what we will do is that we will now transfer these proteins from the gel onto a solid support, which will be a membrane. So what is the necessity of membrane here? The membrane will help in detection of the protein in the subsequent steps. So this is the transfer process. So the membrane which is typically used is PVDF or nitrocellulose. So what is done in this case is that the membrane, which is this one, is placed on top of the gel, directly on top of the gel, and then for support, filter papers are kept on top and the lower side of it so that they remain squeezed tightly, and then they are placed in this gel holder cassette, and finally this gel holder cassette is placed here into this buffer tank by filling with transfer buffer, the buffer in which the transfer will take place. So this transfer buffer typically contains tris, glycine, and methanol. So in this buffer, this gel holder cassette is placed and this buffer tank is filled with this buffer and electric voltage is applied. So now what happens? The proteins are negatively charged because of the application of SDS, right? So now these proteins which are negatively charged will try to move towards the anode, which is positively charged because of the application of the electric voltage. And the membrane side of this cassette is placed at the side of the anode in such a way that when this electric voltage is applied, the proteins from the gel will move towards the membrane. So this process is actually a wet transfer method. There are several other transfer methods like dry transfer, semi-dry transfer. The wet transfer is the most commonly used method, and it also takes a little bit more time than the other two methods. So by this process, the proteins from the gel are now transferred onto the membrane. So next step is our detection or visualization. So now all the proteins are transferred onto the membrane. So now we have to detect our protein of interest. How do we do that? For that, we use antigen antibody interaction. And as we know that this antigen antibody interaction is quite specific, so this is the method that we use to detect our proteins also. So this detection is actually a multi-step process. So the first step of it is blocking. Blocking is done to minimize non-specific binding and non-specific so that the antibodies which we will be using in the subsequent steps do not non-specifically bind to the membrane. So suppose our membrane is like this. So there are many proteins. So these are the proteins which are now onto the membrane after the transfer, and these red ones are our protein of interest, right? Now, what we need to do is we need to block the empty spaces that are there in the membrane. So we use blocking agents which can coat these empty spaces on the membrane so that any antibody cannot non-specifically bind to the empty spaces on the membrane. So there are different blocking agents that we may use. The most commonly used ones are skimmed milk and bovine serum albumin or BSA. Skimmed milk contains many proteins, but bovine serum albumin is a purified protein which we also may use for blocking. Now, after this blocking step is done, we wash the membrane very well. So washing is done by buffer TBS or TBST. So TBS is basically tris and NaCl. And there is an extra T in TBST which stands for a detergent, a very mild detergent called Twin 20. So this wash has to be very carefully done so that the excess blocking reagents can be removed. So the next step is incubation with primary antibody. Primary antibody is the antibody which is specific to our target antigen, and the antigen here is the protein of interest. So now, after blocking, what has become is that this membrane is there which is blocked with non-specific proteins, and there is our protein of interest also. So now what will happen when we incubate this membrane with primary antibody? Primary antibody will come and bind to this protein of interest. So the membrane has to be incubated with primary antibody for some time, and then it has to again be washed so that we can remove the excess primary antibodies which are not bound. Next step is the incubation with secondary antibody. The secondary antibody can be either enzyme conjugated or fluorophore conjugated. So what is the necessity of this enzyme or fluorophore? They will help in detection. So the detection process can be of various types. We can in various ways detect the protein of interest. The commonly used method is chemiluminescence. The two other methods are colorimetric method and fluorescence method. So if the antibody is conjugated with the enzyme, which either gives a colored product or a chemiluminescence signal, then we will be able to detect it accordingly and image it. Or we might also use an antibody which is fluorophore conjugated. So when the secondary antibody will interact with the primary antibody, it will give a particular fluorescence signal. So this is how the detection process is completed. And now we will see a typical Western blot image, how it looks like. And this is how an image looks like. So from different cells, cell lysates are taken, and with the help of anti-tubulin antibody, tubulin protein is detected from different cell lysates. These are the different cell lysates where this tubulin protein is detected. So Western blot can give us qualitative information or also semi-quantitative information about the expression of a different types of proteins. Qualitative means whether the protein is present in the cell or not, and semi-quantitative means we can get an idea about the relative abundance of the proteins, whether the protein is expressed more or whether the protein is expressed less. So that's all about Western blot. If you like my video, please like, share, and subscribe. Thank you.